All right, I'd like to thank the organizers for this opportunity to talk about some of the CEST studies and how we do these technically at our site, both at Johns Hopkins and also Kennedy Krieger Institute. So I always like to start off with kind of a cartoon to depict what CEST is. And so this is our cartoon that we like to show. You have CEST agents are, in general, you have an MRI pulse sequence here. We add this preparation period where we create the contrast, and how we do it is the following. You use this long saturation pulse, which you place on resonance with exchangeable protons. As the signal is destroyed, the exchange transfers these protons, signal loss to water, and so you see this buildup of water signal loss introduced by this saturation pulse. And so if this exchange is very fast, you can get very, very large amplification factors on the order of thousands for this proton onto water. And so it's an amplified contrast mechanism. The first papers describing this effect came out in the early 2000s, both at NIH, Torino, UT Southwestern, and also our site. And since its introduction, the term being coined in 2000, it's grown every year. People have applied this types of imaging to stroke, brain tumors. First patient studies using CEST imaging occurred in about 2006. We had reporter genes, and then we had, more importantly, discriminating necrosis of the brain from tumor recurrence. And now we actually have a full textbook, 460 pages, describing all aspects of this field. And so when you think about CEST experiments, what you have to do is you have to think about sweeping this saturation pulse across the spectrum. So it's a little bit similar to spectroscopy, a little bit similar to CW spectroscopy. You move this saturation pulse. When you get on resonance with the labile protons, you see very large losses in water signal, and then they recover. And so when you plot this water signal as a function of frequency, you'll see this type of pattern where the CEST effects are here. And so what we typically do is we subtract the left from the right side of the spectrum to isolate this CEST contrast, although there are other approaches. And so one of the first applications of this was in brain tumors. And so why was that the case? Well, when people were doing spectroscopy, including Peter Van Zyl and Suzuma Mori at our site, they were looking at these perfused cells in NMR tubes, and they noticed when they switched from standard jump return water suppression to a gradient-based water suppression, that all of a sudden these large amounts of amide protons hopped up in brain tumor cells. And so this basically motivated them to try and place a saturation pulse here and then look at the water signal to do the inverse experiment and detect the tumors. So after that discovery, they moved on, tested on rodent brain tumors in 2003, showed a nice CEST effect in the tumors compared to the contralateral healthy tissue. And then within 3 years, they actually did the first studies on our clinical scanners, the Philips scanners at 3T, and saw very nicely that the high-grade tumors would in fact enhance. So how would you do this type of experiment? Here's just a simple test of CEST imaging using a bunch of phantoms. So you can place many phantoms in here. This is a phantom where there's a lot of air glass interfaces, so the shimming will not be so great. You can get images like this using this type of collection of samples. And so when you look at the CEST contrast or MTR asymmetry maps for this type of phantom, some tubes will highlight, others tubes will not. Those you would expect that the tubes highlighting have in fact CEST effects. Unfortunately, with this type of phantom, there's a gradient of maybe 500 hertz in B0 fields, and so some of these tubes are actually falsely creating the appearance of CEST contrast. And so what we have to do is we have to do use some method to correct the B0 inhomogeneity and then extract the correct CEST contrast from it. So we have to have some way to shift the water resonance. And so the way we did this before is we had our CEST contrast spectra, and then instead, to map the B0 field, we used a much weaker RF saturation pulse. And what this does is it allows us to get a very nice, tight definition of where the water peak is. This separate B0 acquisition, which we termed Wasser. And so we like this. We use the same exact acquisition scheme, so the same artifacts that are present in the Wasser B0 maps are present in the CEST images. And so it's a very nice, simple method for mapping B0. And so from a technical standpoint, we actually can do in any number of ways to fit the data. We either use Lorentzians or higher order polynomials or spline interpolation of the Wasser spectra to create these B0 maps. Here's just a Lorentzian expression here. And then we take our high power data and then we spline interpolate using the B0 determined on the maps and shifting and correcting the frequency that way before we calculate the asymmetry. So here's an example of an uncorrected MTR asymmetry, and then after our Wasser suppression, you see that you have pretty healthy preservation of the features of the CEST contrast. As you get really close to water, you can have a couple % errors, so this does occur. So that was a very simple example. How are actual in vivo apt contrast studies performed at our site on clinical scanners? And so I should like to point out that Philips Healthcare has spent a good bit of time optimizing their 3T clinical scanners for CEST imaging. Here's a slide that Jochen Kuiper kindly provided to describe some of this. So on their clinical 3T scanners, they have a parallel transmit channel. It has two separate transmit channels. And this is very nice for CEST imaging because you have two different sets of RF amplifiers, one powering the one channel, the other powering the second channel on the body coil, which is used for transmit. And then what you can do is you can flip as the amplifier starts to droop, which occurs about 250 milliseconds after the start of the saturation pulse, to the other channel and hop back and forth. And what this does is this preserves a nice flat saturation over several seconds, which is what's excellent for CEST imaging. So Philips spent some time developing the software and the hardware to perform this RF hopping. And so they use separately. They use the receive coils. Any number of receive coils can be used. They shim both the RF amplitude and phase to try and get homogeneous between the two channels, and so that they have some extra features in terms of making sure that both channels have very nice B1, which is similar between the two channels. And so in their general case for brain tumors, they use a fast spin echo sequence with a 120 degree refocusing pulse, centric encoding, and they can achieve up to 4 microtesla saturation powers. Typically for apt, you use about 2. And here are some other parameters. So for their apt experiments on patients, we like to try and keep the time down. So in this study, we actually do just a 9-point collection. So these are 4 points here. This center point for the where the actual contrast is maximal, is acquired 3 times with 3 different echo times. And the reason they do this is to acquire a B0 map just using these different echo times, the signal phase differences. And so on these Philips scanners, they shift the echo over the 3 acquisitions and then use a 3-point Dixon method to basically create the water, fat, and B0 maps from the first signal in the echo train. And again, here's some very nice images. For the high-grade glioblastoma, you see very, very nice CEST contrast maps, which are different than the FLAIR images and allows you to really see quite nicely the high-grade tumors. You can also differentiate between recurrent tumor and pseudoprogression. They've shown this in some other studies. And also now at our site, we even use, they've integrated these CEST images into the brain lab navigation system to allow the neurosurgeons to actually use it for guiding the needle biopsies. And so they've seen some improvement in collecting very well cellular samples using this apt imaging. So there's some controversies when you talk about amide proton transfer. You have this type of shape, but of course there's multiple contributions. It's a biological tissue and it's an endogenous contrast. There's not one source of it. And so some people have been discussing whether or not there's some influence from the NOE effects compared to the amide protons. At our site, we generally just don't worry too much about that. We use the asymmetry, which is a very simple metric, and just see whether or not it can be used to differentiate high-grade tumor from low-grade tumor, and also from radiation-induced necrosis. And also, separately, other sites will use different B1 powers, and so there can be slight differences in terms of the amount of this enhancement and whether or not it's the amount of the amide proton contribution compared to the NOE contributions. And so there's some need for scanner companies to standardize, come in and help standardize this. So that's for just looking at brain tumors with amide proton. In addition to excess mobile proteins, there's also other metabolites that are altered in cancer cells. And of course, this has been studied for a long time with magnetic resonance spectroscopy, which has allowed the detection of different metabolites, particularly the cholines. However, there's major sensitivity limitations in MRS studies. And so what we were interested in is trying to look at whether or not we could detect metabolic differences in breast tumor cells using CEST. Now, one of the features of CEST is that because of the fast exchange, the proton lines are broadened. It's a T2-related broadening, and so there is some overlap which occurs between creatine, myoinositol, glutamate, glucose. These are a couple of the main metabolites that you might see in tumor cells. However, there are also different exchange rates. So what we've noticed is that when you look across the spectrum, you can see differences in tumor aggressiveness based on looking at these CEST metabolite profiles. This is something we published in NMR on Biomedicine, and we're working now towards trying to separate these metabolites. Now, I'd like to also mention, as a technical note, there's some complications when you talk about CEST imaging in breast cancer compared to in the brain, because there's excess fat signals from water. So these fat signals here can contaminate the Z-spectra or MTR asymmetry profiles. And so for this case, this fast spin echo Dixon, 3-point Dixon method is very nice because you can then separate the water only from the the multiple signal sources, which includes fat. And so this is a very nice thing that was published recently, that using water only is much better and cleaner for looking at the metabolite profiles in breast cancer. So, okay, I talked about endogenous contrast. I'd like to also talk about the case where we would administer a CEST agent as a contrast agent. So what type of sequences do you use for that? And when I mention that, I should describe what a contrast agent is. Oftentimes, we need to administer some sort of exogenous entity to alter the MRI signal and highlight better an anatomical or functional region. And of course, this is performed in about 30% of all MRI scans to highlight pathology, particularly for brain tumors and also breast tumors and multiple sclerosis studies. And people have long used gadolinium, but of course, Henrik Thompson and coworkers noticed that for patients with bad kidneys, nephrogenic systemic fibrosis can occur, which is associated with gadolinium administration. In addition, with a number of different administrations of gadolinium, this gadolinium will accumulate in brain tissue over time, which raises safety concerns. And so there are some interest in developing alternatives to gadolinium. So there's many different options for exogenous CEST agents, the different shift, different exchange rates, and then you can have like agents with just 1 or 2 protons, and then you can have agents with multiple protons with different pH dependence to their exchange, agents such as iopamide. And so one of the first patient studies that we've performed at our site is actually administering just dextrose or glucose. And this is the idea that sugar is a nice biodegradable contrast agent. So when we administer glucose, we'll use a different type of approach. We'll actually just look at a single saturation frequency instead of multiple to create an MTR asymmetry. So just look at the signal at 1.2 or 2 ppm from water. And then this also folds in T2 and CEST mechanisms, so it's not a single pure source of contrast. And might need to discard the first group of agents before a steady state is established. And we can also use area under the curve analysis. So here's an example at 7T where a patient had a brain tumor. Here's the gadolinium T18 weighted image, and we infused glucose and looked at the uptake. Here's a plot showing how the artery glucose behaved, and the tumor rim and tumor core. You can see the tumor highlight here with our first patient study on glucose imaging. Here's another patient with a recurrent left frontal glioblastoma. At the early time, here's the gadolinium contrast image. At the early time points, you can see that the tumor rim is highlighted, and then it grows in more over time if you look at the later saturation images. And so we've shown that we can actually detect this glucose enhancement in patients at 7T and 11/7T. Now, when you go down in field to clinical 3T scanners, the signals coalesce because of the speed of the exchanges the rate of exchange is much higher compared to the chemical shift difference. So you have more broad signals, more T2 contrast. And so in this case, it's really important to fold the T2 and the CEST effects in using this just a single saturation image compared to 11.7 where you can just look for the pure CEST signal. And so here's just some first study of an infusion of a patient with brain tumor at 3T. You can see a very nice highlighting in the area of the tumor through the saturation signals. Here's a comparison between the gadolinium DCE image and the glucose image. and the flare. So it's also looking not exactly the same, but having some of the same features. And here's just another example where you can use a spin lock pulse to do something the same, again highlighting this and also the blood vessels nearby. And you can also use on-resonance binomial pulse labeling to perform this type of study. So with that, I'd just like to conclude. Previous. So CEST MRI is a new tool with utility in a wide range of medical applications, and the protocols will be different whether or not you're looking for endogenous contrast or administering an exogenous entity. And after the profound success seen in the brain tumors, as I showed from the biopsy and treatment, the methods are now migrating to other medical applications. And just like to point again that we do have a textbook if you're interested in more details on all the different types of CEST imaging studies. So thanks very much.